This is a video abstract of Edinburgh's 2012 iGEM project. This year, we are working on 3 projects. We would like to make a bioelectric interface, characterize the bacterium Citrobacter freundii as a chassis, and make new antibiotic-independent selectable and counter-selectable markers. The aim of our bioelectric interface project is to enable communication between electric devices such as computers and biological entities. To enable communications, we want our bacterium to release electrons in order to talk to computers in a common language. E. coli can't normally do this, but another bacterium, Shewanella onadensis, can. What we want to do is take the MTRC-AB genes from Shewanella and express them in E. coli. These genes enable the transport of electrons out of the cell. We also need the CCM genes, which are responsible for cytochrome maturation. Even though they are native to E. coli, they are expressed only under very specific conditions, so we want to link their expression to an inducible promoter. The CCM gene products help with the maturation and folding of the MTRA and MTRC cytochromes. They also help transport these to their final destinations in the periplasm and outside the cell respectively. Once everything is in place, the system will take electrons generated by the TCA cycle and ship them to the cell membrane via an electron transporter called quinol. The electron will then move from protein to protein, starting with the native E. coli protein NAPC in the inner membrane, to the MTRA protein in the periplasm, and finally to the MTRC protein outside the cell. The MTRA and MTRC proteins are held close together by MTRB, which is a protein that forms a channel in the outer membrane. Ultimately, the electrons will arrive on the electrode to be detected by an electronic device. This will establish a connection between a biological and an electric system. Our second project involves the bacterium Citrobacter freundii, the friendly lemon bacterium, which also wants to be part of iGEM. We want to look into its suitability as a chassis by characterizing its compatibility with the biobricks that were made for other organisms. Its advantages are that it can grow in salt water, so it could be used for bioremediation, and we can grow it without wasting drinking water. It is also less associated with diseases in humans. Finally, we find its name to be more appealing. To further characterize it, we want to look at its genome and compare it to E. coli in order to find how it differs from it, and whether it has things that could make it a more suitable chassis. Does the fact that it is less associated with disease in humans make it safer to release into the environment? The answer is no, because any novel genes inserted into it will probably be selected for with antibiotic resistance markers. Whenever an organism is genetically manipulated, we want to differentiate between the cells that have taken up DNA and those which have not. This is normally done by including an antibiotic resistance gene on the DNA insert, so that the cells that did not take up the DNA will be killed by the antibiotic included in the growth medium. However, releasing engineered bacteria into the environment may result in the spread of antibiotic resistance in the wild bacterial population. This is why we want to develop non-antibiotic selectable and counter-selectable markers which will eliminate this risk. Ultimately, we want to make genetically modified bacteria safer to release into the environment, while also enabling communication between the biological and the electrical worlds.